Today is seminar number 4 in the series. We've had 3 so far, starting with the origin of the eukaryotic cell and updates to the tree of life. Last week, we heard some great talks on the hidden biochemistry within microbial dark matter and their role in biogeochemical cycles. Today's topic is cultivation of new lineages. A very quick outline of today's proceedings is that we'll have 2 invited speakers followed by a very short Q&A session for each of those speakers. Please put your questions in the Q&A feature that Zoom allows at the bottom of your screen. We will also have 2 additional short format flash talks. However, there'll be no Q&A for the flash talks, but all presenters will be available in the breakout session after the seminar. So without further ado, it is my pleasure to introduce today's moderator, Dr. Mircea Podar, who is a distinguished staff scientist and leader of the Systems Genetics Group at Oak Ridge National Lab. Take it away, Dr. Podar.
Good afternoon, everyone, or good evening. Welcome again to this very exciting topic on cultivation of novel lineages. We can look at them like the opportunity to meet new life forms, and we have some very exciting talks scheduled for today. We are going to switch to our first flash talk, who is going to be by Dr. Marike Palmer. Marike is a postdoctoral fellow right now with Brian Hedlund at the University of Nevada in Las Vegas, where she studies she's interested in mixing genomics, phylogenomics, genetics, and microbial ecology to look at the variety of microbial lineages. And Marike did her PhD and master's in microbial systematics at the University of Pretoria in South Africa under the guidance of Fanus Venter and Emma Stielkamp, where she graduated in 2019. Welcome, Marike.
Thank you so much. First off, thank you to everyone for the opportunity to actually talk about this work today. And I'd like to start off by saying I don't think we need an introduction to how difficult it actually can be to cultivate these novel lineages. But today I would actually like to discuss with you a project that involved a fortuitous isolation of these novel organisms in the fact that they weren't the target organisms for isolation at all. So if we just look at a quick overview of the, there we go, of the actual workflow that was used. So this specific project originated through isolation efforts for this project indicated in this paper that came out last year. And overall, it entailed sampling of sediment and microbial mat and water samples of 2 hot springs, one in the USA and one in China, and a reasonably straightforward isolation procedure was used for these organisms. But in 2 of the isolates cases, these turned out to actually not be thermos species. So having the isolates in hand, we actually set out to sequence the genomes of these organisms. And the first one was sequenced through a CSP project through the JGI. And the other one was sequenced at Novogene in China. And we found out that both of these organisms for which we had genome data and 16S data now available actually represented novel members of the Chlorophlexota or the Chloroflexi. So in the meantime, a novel class within the Chloroflexota was described, the Teperiformia, and the genus that was the type for this class is Teperiforma, with the type species for this genus being Teperiforma bonchosmolosskaya. And having the genome sequence for this organism and then our 2 new isolates, we could actually identify that both of these isolates represented new species within the genus Teperiforma. But at this point, we were still somewhat uncertain regarding the actual the most appropriate taxonomic rank for these organisms. So for this purpose, we decided to use all of the available high quality Chlorophyllexota genomes, and we reconstructed a phylogeny for the phylum using the full alignment for the BAC 120 dataset, where we partitioned the concatenated matrix and applied the appropriate model to each of the partitions. And based on relative evolutionary divergence and this robust phylogeny that we actually got really good support for the different classes. And based on this data, we could actually identify that the most appropriate taxonomic rank for this group of organisms would be an order within the class Dehalococcoidia. So for those that are not super familiar with the Dehalococcoidia, this class is basically most well known for the organohalide respiring microbes within this class. And overall, it's typically grouped into terrestrial clusters or marine clusters. And in the terrestrial clusters specifically, many of these microbes are considered really important, especially for potential bioremediation. Whereas the marine clusters are really important and abundant in the ocean. But overall, this class is quite poorly understood. And in this entire class, there are only 2 other genera that actually contains cultivated representatives, particularly in pure culture, and both of these genera actually fall within the previously identified terrestrial cluster. So obviously, having these isolates now in hand, we wanted to find out more about these organisms, but the ease with which they were isolated definitely were not transferred to the ease of working with them in the lab. And to date, we've actually still been unable to cultivate them successfully on defined media. So trying to understand more about these organisms, we decided to employ alternative approaches. And the first approach we used is Exome Metabolomics. And this we used, this we did in collaboration with the team at JGI. And what we have on these graphs over here, we've got basically the substrates below this line and the products being produced above this line. And what was really striking and very much unexpected from this data is that there was a lot of aromatic compounds that seemed to be metabolized. But there was also some accumulation of really closely related substrates that occurred during growth, so specifically related to these aromatic compounds, which kind of led us to believe that they're either somewhat inefficient in the degradation of these aromatic compounds, or that they might be involved in some form of consortial degradation. We then decided to look at stable isotope experiments. And when we're looking specifically at the C13 compounds being respired, we could see that acetate pyruvate and ribose showed high levels of respiration, whereas the C13 incorporation again showed for acetate, pyruvate, and ribose, but also for a couple of other compounds. But really strikingly and very much surprisingly and unexpectedly, we actually saw that a few cells had very, very high levels of incorporation from C13 lignin. And this is something that we're still looking at and still performing experiments on, but this is quite an exciting finding, particularly in light of the fact that Teperiforma bonchosmelovskaya has been shown to grow on lignin. So this is definitely something that we're following up on. So of course, the further phenotypes that we wanted to look at, so particularly either predicted from the genomes or otherwise observed, entailed firstly looking under the microscope at these organisms. And as with the previously isolated member of this genus, we could actually see that the cells had these really interesting conical poles, but very much unexpectedly as well, we saw flagella in many of these micrographs, as well as a peptidoglycan layer. And today I'll specifically focus further on the flagellum-related data. So when we look at the flagellar biosynthesis genes, we, of course, having the genome, like really, really good genome representation for this class allowed us to actually look at the distribution of these genes in these different organisms. And we decided to do an ancestral character state reconstruction from this data. And also, surprisingly, it appears that the biosynthesis genes that were present within the genomes might actually be an ancestral trait within the class. And we looked at the flagellar gene clusters as well. So consistently, we were able to identify 3 main clusters within the genomes that actually had these genes. And if we're looking at specifically the genomes that were assembled to replicon level, it seemed that these 3 gene clusters are somewhat scattered throughout the genome. But the synteny of those gene clusters seem to be reasonably comparable and really highly synthenic. So at the moment, we're busy looking at the evolutionary history of the different gene clusters by specifically doing phylogenetic analyses on these genes identified in green over here. So if we're just summarizing all of the findings and some of the future questions, we've been successful in isolating 2 novel members of the Dehalococcoidia, and these members seem to be capable of motility through flagella. And this would actually be the first report, as far as we're aware, within the Chloroflexota, whereas the peptidoglycan biosynthesis, again, really unexpectedly, might actually be the first observation within the Dehalococcoidia. And again, this is the 3rd genus that actually contains cultivated representatives within this class. Another really interesting aspect that we're definitely following up on is aromatics degradation, and particularly lignin and lignin-derived aromatics. And with that, I would like to thank all of the contributors to this work, and thank you for your time.
Thank you, Marike. As we mentioned, there's no time for Q&As right now, but please stay after all the talks are finished for the breakout session, and then you can ask Marike questions. So let's move to the second speaker from the short talks. I'd like to welcome Dr. Steffen Buessecker. Steffen is a postdoc in the Decas lab at Stanford, and he studies thermophilic communities in the Great Boiling Springs in Nevada, focusing on tungsten utilization in Caldarchaeales and on methane cycling. He completed his PhD at Arizona State, working on nitrous oxide cycling in tropical forest. Steffen, please.
Thank you so much for the introduction. Yeah, I'm really happy to be here today and to be given the chance to talk about this really great project. This is my first postdoc project coming to Stanford. So I'm really excited to talk about this. And right at the beginning, I wanted definitely to make sure to give credit to the initial work that was done, that this work is based on, and that is those are undergraduate researchers of the Jeremy Dodsworth lab at CSU San Bernardino, which did fantastic work to enable this project in the first place, to get a novel Aigarchaeota microbe into culture that we later could then look at. And this is a really big project too. So we have different collaborating universities, and I'm really lucky to be part of this big project of so many people contributing. When we think about tungsten and the biochemistry of tungsten, then we can also think about deep time and the conditions, how they might have been on the Archean Earth, because trace metals that are used in enzymes of modern organisms might have been more or less abundant under different conditions on the Archean Earth or at other stages of our planet. Tungsten specifically might have been more abundant, especially in very sulfidic systems, in hydrothermal systems, but also in the oceans of the Archean Proterozoic transition period. And under sulfidic conditions, tungsten is much more abundant than molybdenum that has comparable properties in the biochemistry. So the use of tungsten as a trace metal and using tungsten in tungsten enzymes, because tungsten has 3 oxidation states from 6, 5 to 4, might be a relic from earlier microbial habitats, really. And that is where I'm basing my talk on, in a structure that I first talk a little bit about the enrichment and the dependence of this on this trace metal, and then on tungsten enzymes, and finish with the metabolism of a novel species that shows this tungsten dependence. The first steps to obtain this Aigarchaeota culture was done at the Great Boiling Spring in Nevada, which is a hydrothermal, a hot spring in Nevada, about 85 degrees Celsius hot in the main pool. It has a circumneutral pH. And first there were cornstarch in situ enrichments done that were incubated for about 6 months in the field to enrich heterotrophic microbes. And these enrichments were then transferred into laboratory cultures. The initial enrichment constituted different taxa, and one of them was these Aigarchaeota member, and these members are all anaerobic, and this member was in an abundance of about 3%. It was stable in this in situ enrichment, and this enrichment was then transferred into media of a laboratory culture, and under different conditions, this member showed different abundances. When in general in those media, the Aigarchaeota member was about present between 0.5 and 7%, but it was stable over several transfers if hot spring water was added to the media. In those media compositions where the medium was not amended by hot spring water, the culture lost this Aigarchaeota member, and it did not recover. So the question really was, what is the essential component of the spring water that enables those microbes to grow and to be stable? And at this point, we did 2 things. First, we compared the composition of the spring water to the synthetic medium, and looked at which elements are actually present or not present in one or the other. And we quickly noticed that it might be tungsten, because tungsten was present below detection in synthetic media based on the trace metal mix that we used, but it was present at about 300 nanomolar in the spring water. And since we used a 1-to-1 dilution, it was about 150 nanomolar in the medium with the spring water amended. So that was the crucial difference. And after that, we looked at a metagenome assembled genome from this member, and noticed that there are several tungsten enzymes and tungsten transporters present in the genome. So based on this genome informed approach, we hypothesized that this microbe needs tungsten essentially to grow. And from now on, I'm also calling this Wolframiraptor gelahensis. I will not go into detail how we delineated this species, and that is part of an article that is hopefully soon coming out. But here I'm just calling it in the species name that we designated it. And Wolframiraptor gelahensis is only stable or is only present in the mixed culture with added tungsten concentrations. So in the above panel, you see that there's amplicon sequence variants that show dropping abundances when tungsten is not present in the synthetic medium, always presence when there's spring medium amended, and also the ASVs of Wolframiraptor are present when only 100 nanomolar of tungstate is added. And this is also consistent with the qPCR analysis on the lower left panel, which corresponds to those 3 replicates that you can also see in the ASV panel. And we see a stark contrast that when tungstate is left out of the synthetic medium, that we have very low numbers of this species, whereas this effect is alleviated by the addition of tungsten. And over several transfers, we see on the lower right side that tungstate is essential over several transfers, no matter which substrate, if constover or a sugar mix is added as a carbon source. We basically lose the Wolframiraptor gallicensis species if not at least 20 nanomolar of tungstate is added to the medium. So the oxidoreductases, the tungsten enzymes were, many of those tungsten enzymes or the predominance that we observed in the genome were tungsten-dependent oxidoreductases. And we wanted to look further into those oxidoreductases and understand what they're actually doing in the metabolism of this species. And in the left panel, you basically see an alignment of the sequence of the protein sequence alignments of those oxidoreductases. And you see that in purple, those are coming from the Wolframiraptor gallicensis organism. And they're basically scattered over this tree of oxidoreductases, of tungsten-dependent oxidoreductases. These are amended with reference sequences from known ORs. And you see that there's AOR-like, aldehyde ferredoxin oxidoreductase-like groups in purple. But then you see also very faint some sequences in purple in the 4 area or in the GAP4 area. Complementary to that, we did also a protein structure modeling using the ITESA server and the DALI server, and a multidimensional scaling approach to not only compare the sequences of the oxidoreductases, but also the structures. And the results were very consistent that the ORs from Wolframiraptor gallicensis are AOR-like, but also including GAP4, which is a glyceraldehyde 3-phosphate oxidoreductase, and a formaldehyde oxidoreductase, but all of them are tungsten dependent. So this basically told us that there's the propensity of the microorganism to oxidize a wider variety of aldehydes. And now the question was, where do those aldehydes derive from? What are really the carbon sources? And to answer this question, we used a fish nanoSIMS approach to identify the microorganism Wolframiraptor gallicensis in the mixed culture by fish, and to track uptake of carbon from carbon substrates and the uptake of 15N from ammonium as a general tracer of anabolic activity. So we basically added those compounds into corn-stover mixtures to see which compounds might be preferably taken up. And we see that amino acids and ribose show lower anabolic or induced lower anabolic activity. There was generally always growth, so the mixed culture was growing, which you can see looking at the 15N atom percent excess values. But we see definitely a preferred uptake and an enrichment of 13C and 15N when xylose was added, and this basically made us think that xylose is really the preferred monomer, a carbohydrate monomer of Wolframiraptor gallicensis. This could be in the spring. It could be supplied from exogenous sources, from plants that are growing around and where DOC basically leaches into the spring, or by microbial mats that grow there and fix organic carbon. So xylose could be supplied to the community in the spring by several sources. Now, what is the mechanism of really this tungsten starvation? And we think that xylose-like sugars are taken up are metabolized via a pentose phosphate pathway, then an archaeal Embden-Meyerhof pathway producing pyruvate. This pyruvate is further oxidized, decarboxylated by CoA, where we get our acetaldehyde. Alternatively, there could also be a glyceraldehyde 3-phosphate coming from the ribose. But basically, we're ending up with aldehyde groups from pyruvate. And this aldehyde can then be oxidized using an oxygen atom from water to a corresponding carboxylic acid by AOR and GAPOR, or also A4. And the tungsten is coming from the surrounding of the cell, which is, and then it is brought into the cell by a tungsten uptake protein that is very specific for tungsten. So Wolfram Aigarchaeota does not have molybdenum uptake complexes. So it's specialized for uptake of tungsten, synthesizing tungstapterin as a cofactor, introducing that into AORs, and then AORs oxidize the aldehyde groups. They reduce the ferredoxin, which is fueling a ferredoxin recycling. And this reduced ferredoxin is then used by a membrane-bound hydrogenase, as we believe, to merge two protons to a hydrogen and to induce a proton motive force. And this proton motive force is ceasing if the ferredoxin is not recycled, and the cell would basically run out of energy. And we think that this is a model of that would explain why they don't grow in the absence of tungsten. In summary, the anaerobic Aigarchaeota we culture in media with tungsten as an essential component that has to be present at about 20 nanomolar tungstate concentration. And with this approach, we have this Aigarchaeota culture or stable since over 4 years, transfer every about 3 weeks under anoxic conditions. And Wolfram Aigarchaeota contains these aldehyde ferredoxin oxidoreductases that likely oxidize the wide array of aldehyde compounds. And the mechanism is basically that tungsten scarcity inhibits aldehyde oxidation from xylose-like sugars, resulting in this slower ferredoxin recycling, and therefore a lower activity of membrane-bound hydrogenase and cessation of the proton motive force. And at this point, I would also like to really thank all these great people, and also Dave and Sandy Jameson from that always give us access to the Great Boiling Spring, which is so important to our research, and all the other people that contributed here to the funding agency, NASA Exobiology. We are really grateful here at Stanford and Dikas and myself that we can collaborate with so great people. Jeremy Dodsworth at CSU San Bernardino, Xavier Mayali, Jen Patridge, Peter Weber at Lawrence Livermore National Laboratory, and Marike Palmer and Brian Hedlund at University of Nevada, Las Vegas. And thanks again for this great chance to present our results here at the NeLLi Symposium. Thank you so much.
Thank you, Steffen. Again, please hold your questions for the breakout session. I'd like now to introduce Dr. Purification Lopez-Garcia, who is a research director at the French National Center for Scientific Research, CNRS, at the University of Paris-Saclay. Puri has a long list of accomplishments and interests. She has contributed significantly in the study of the origin of life, early evolution, extreme environments. She is a member of the American Academy of Microbiology. She was also elected in the Academia Europea, the Académie Royale des Arts et Sciences de Belgique. In 2017, she received the CNRS Silver Medal, and she has a long list of involvements in various scientific advisory boards all over the world and various exobiology institutes, for example. And I will not take from her time. Puri, please go ahead.
Thank you very much, Mircea. And so good afternoon to everybody, and thank you once again for this invitation to this to present our work on vampire bacteria in this NeLLi symposium. So you are all familiar with this representation of the tree of life. This is a phylogenomic tree based on ribosomal proteins encoded in the genomes of diverse culture microorganisms, but also in sacs, genomes amplified from single cells, and MAGs, genomes assembled from metagenomes from environmental sequences. These studies confirm and extend actually previous observations based on 16S ribosomal RNA amplicon sequencing, showing that the vast majority of the microbial diversity belongs into groups that lack cultural representatives so far. These are all these red dots here. So this is particularly striking in one group. There is this large clade, the candidate phyla radiation, CPR bacteria or Patesi bacteria, that can encompass up to 25% of the known bacterial diversity. The genomes of these bacteria are reduced and encode for very limited metabolic activities, and this strongly suggests that they strictly depend on their host for a living. And these seem to be actually validated by the first description of Saccharibacteria maintaining culture with their host. They are these epibiotic bacteria parasitizing actinobacteria. They have tiny cells, so the scale here is 500 nanometers, and this actually corroborated previous microscopy observations of potential CPR cells in nature. So the CPR bacteria in this tree appear at the base of other bacteria, at the base of the bacterial branch, and these seem to suggest that perhaps these limited metabolic capabilities were ancestral. However, more recent studies, more thorough phylogenomic analysis now include clearly the CPR well nested within the Terrabacteria, one of the two large groups that are comprising within bacteria. And so this suggests that on the contrary, CPR are more derived bacteria that possibly reduce their genomes and their metabolic capabilities from more complex ancestors. And these likely accompanied an adaptation to parasitic or perhaps other types of symbiotic lifestyles. But this is still hypothetical because we only had until very recently the example of Saccharibacteria. So what is the biology of the other CPR lineages? Here I'm going really to start my story that comes back 40 years ago, or back in the time, to this particular place, a small sulfidic lake in Catalonia in Spain, Lake Siso, which bears blooms of anoxygenic photosynthetic bacteria that give this pinkish color to the waters from time to time. And so in 1983, Isabel Esteve and her co-workers observed for the first time this type of epibiotic bacteria on cells of Chromatium minus, which was the dominant anoxygenic photosynthesizer in these blooms. And a bit later in 1986, Ricardo Guerrero from the same group in collaboration with Lynn Margulis described the genus Vampirococcus to refer to these parasitic cells that actually they qualified as predatory because they ended up by killing their host and emptied the cytoplasm of these photosynthetic bacteria. And they made beautiful scanning and electron and transmission electron micrographs that you can see here for the time. And in these cells, you can see clearly that the parasite, or this Vampirococcus, forms piles of of cells forming this type of stalks that are attached to the photosynthetic bacterium. Unfortunately, at the time, it was not possible to identify this organism by molecular sequences, and the story remained there until relatively recently, when we started to study microbial mats from this lake, the Salada de Chiprana, also in Spain. It is a saline lake having between 4 and 9% salt, and it harbors conspicuous microbial mats that we are very particularly interested in studying. So they are thick, stratified. You can see here the layering with a green layer of cyanobacteria on top, and then other layers, including these pinkish layers that are enriched in an anoxygenic photosynthetic bacteria. These bacteria are mostly belonging to the Chromatiaceae. These are gamma protobacteria that use hydrogen sulfide that's electron-producing sulfur granules that remain in the cytoplasm in the form of these refringent granules. So and this is what the community looks like under the microscope. So we collected these mats and placed them in mesocosms in the laboratory. And after a few days, we had a look to the water that was surrounding these mats. And we observed blooms of these purple bacteria. And together with that, we observed morphotypes that look very much like Vampirococcus cells. They also they attach to these photosynthetic bacteria, forming these stalks of piled cells. Sometimes they can connect different photosynthetic cells, suggesting that they are very sticky. One photosynthetic cell can be colonized or by several parasitic cells. And the photosynthetic cells that are actively moving and swimming at the beginning progressively become non-motile, and in the end, the cytoplasm change the color, and you end up by having this type of ghosts that look here very dark, with parasite cells still attached. So this clearly indicates, if you observe this mesocosm in the lab, that these Vampirococcus species are parasites that kill their hosts. This is the definition of a parasitoid, and we can consider this as parasitoid predation. We also did some scanning electron microscopy. Here, the host is not very well fixed, but you can observe clearly the parasites. They have around 500 nanometers of diameter and around 100 nanometers in thickness. And they form these piles that were very characteristic of the original Vampirococcus descriptions. So we decided to this time identify molecularly these parasitic cells, know where they fit in the tree of life. And for that, David Moreira in the team micromanipulated individual consortia of Vampirococcus with its host. And all of these consortia yielded the same 16S ribosomal RNA sequence for the host, which was a member of the genus Halochromatium, and the same sequence for the parasite. So this suggests that these are clonal populations and that there is a specificity of the parasite, of this host parasite couple. And of course, we decided to sequence the genome using single cell approaches. After the amplification of the cell, we amplified the genome, applied Illumina high-seq Illumina sequencing to sequence it, and then assemble the genome of the parasite, applying binning approaches to separate it from that of the host, prior to annotation, of course. So the genome of Vampirococcus is relatively small, 1.3 megabases, but it is comparable to the smallest genomes of some free-living organisms. It has the peculiarity that the UGA codon encodes for glycine and is not a stop codon as in other organisms, has only one ribosomal RNA operon and encodes for around 1,200 proteins. By contrast, we could not, so the genome of Vampirococcus is complete, but we could only obtain around 10% of the genome of the host halochromatium, and this actually suggests that the consortia that we pick were actively being, the host was actively being degraded and including its genome by Vampirococcus. So we place Vampirococcus in a phylogenomic tree using proteins, and it turned out that Vampirococcus is nested within this CPR bacteria and is in particular sister to this Abscondita bacteria phylum, the former candidate division SR1, and they are sister to the Gracili bacteria and Peregrini bacteria within this CPR radiation. So with this information, we decided to describe a new species of the genus Vampirococcus, which is actually the first described species. And we call it Vampirococcus lugosi in the memory of Bela Lugosi, the actor who played the role of Dracula in the famous 1931 movie. Around half of the genes encoded in the Vampirococcus lugosi genome cannot be assigned to known clusters of orthologous genes. So we ignore completely their function, and there are many questions about them. So many genes without a known function. And actually, Vampirococcus shares a limited number of genes with the other genomes available for Abscondita bacteria, not even 400 genes. Comparative genomics and the analysis of gene content across these three major CPR phyla, Abscondita bacteria, Gracili bacteria, and Peregrini bacteria, including here Vampirococcus, allowed us to infer the presence of around 1,100 ancestral genes to the whole clade. And then these genes were differentially lost in the different lineages. So Vampirococcus had lost around half of these ancestral genes. And it's likewise, the others also lost a lot of genes. But there was also gene gain, so there are many possibly come from just innovations, but also some genes might have evolved beyond recognition. But there are also genes that have been acquired by horizontal gene transfer, and you can show that clearly by analyzing the phylogenies of all the genes in the Vampirococcus genus. So in this way, we detected 38 genes acquired by horizontal gene transfer by the ancestor of Abscondita bacteria and Vampirococcus, and an additional 124 genes gained in the Vampirococcus line. This is a chart of the metabolic chart of Vampirococcus inferred from the genome, and it is almost empty, only a few green lines. And this actually illustrates very well the limited metabolic or in particular biosynthetic activity of Vampirococcus, no synthesis of amino acids, lipids, vitamin, nucleotides, and other cofactors, etc. And it has no canonical electron transport chain. Vampirococcus is able to produce ATP by substrate level phosphorylation, fermenting starting by glycerate 3-phosphate, which is a product of Rubisco, and it's possibly the metabolite that it obtains from the host in large quantities. And it is relatively different of other Absconditobacteria that can use alternative or other substrates as well. And possibly this reflects also that they parasitize other types of hosts. One of the most important features is that Vampirococcus has a very elaborated cell surface. This can be shown by our TEM pictures, but that are actually very similar to those produced in the 1980s. So Vampirococcus is a monoderm bacteria. It has only a cell membrane, and this actually agrees well with the fact that there are no genes encoding for proteins typical of the outer membrane in gram-negative bacteria. But on this, over this cell membrane, you have a thick layer of fibrous material. And actually another observation is that this type of fibrous material seems to separate Vampirococcus from the host. So there's a thick space here filled by this type of material, and we have been unable to observe any type of cytoplasmic bridge or membrane-to-membrane contact. And so this cover of fibrous material is consistent with the presence of many genes involved in different compounds that can be on the membrane. There is, of course, the peptidoglycan of the cell wall that for which Vampirococcus has all the genes, glycosyltransferases, secretion system, and several type 4 pili, but also giant membrane proteins, all of which tend to have motifs that imply interactions, protein-protein, protein-lipid interactions, and also motifs involving cell adhesion. So this means that Vampirococcus is sticky. It tends to, its surface tend to stick to other cells. Vampirococcus also encodes 2 murine DD endopeptidases, and this is something that it shares with Dehalococcoidia, which is another very famous predatory bacterium. So they both have a self-wall endopeptidase that is involved in the growth of their own cell wall, but they have what we can call a predatory endopeptidase that is likely targeting and degrading the peptidoglycan of the host. And Vampirococcus has, in addition, a domain 3 that is possibly involved in some specificity, which is lacking in Dehalococcoidia, which can prey on different hosts. Vampirococcus also encodes different defense mechanisms that protect the cell against the invasion of foreign DNA, toxin-antitoxin, restriction modification system, and also CRISPR-Cas systems. So they protect against foreign DNA, but they can also help to degrade foreign DNA. And actually, we think that that is one of the activities or objectives of these mechanisms to contribute to the uptake and degradation of DNA from the host. So it has 2 different variants of CRISPR-Cas systems. One of them, this class 2 type 5 CRISPR-Cas system, appears to be conserved in the CPR bacteria, can be deduced from the phylogeny of Cas1 enzyme, which is very conserved in the CPR. But the other CRISPR-Cas system, which is this class 1 type 3, appears to have been acquired horizontally, as this Cas1 enzyme is embedded among other Cas1 enzymes coming from other different types of bacteria. So there are many new genes in Vampirococcus, as I said, but it is very difficult to predict their function because in many cases there are no conserved domains, but there are exceptions. And this is one of them. This is a protein that combines in a peculiar way or original way 3 different domains, one cytochrome B1-like, one proredoxin and one ferredoxin reductase motif domain. And they are assembled in a protein that has transmembrane motifs. So This protein is mostly involved in the translocation of electrons and or protons across the membrane. So even if Vampirococcus does not encode for a canonical electron transport chain, this protein may create a proton gradient across the membrane that can be used by the ATPase to synthesize for ATP synthesis. So with all this information and with the observation of Vampirococcus under the microscope, we can reconstruct the lifestyle and the biology, cell biology and molecular biology of this organism. So Vampirococcus is an extreme parasitic bacterium, a predator that attaches to the host via large giant proteins and glycoproteins, and it then releases enzymes and other mechanisms and uses other systems to create holes in the cell wall and the membrane of the host. And then it releases various hydrolytic enzymes to this fibrous space that separates the parasite from the host. And most likely from that limited diffusion, Vampirococcus can acquire or take via a variety of transport systems different metabolites into its cytoplasm for its benefit. So although we can describe the biology of this organism from this data, we actually do not keep Vampirococcus in culture because Vampirococcus is a highly virulent predator, and actually it kills the host very rapidly and it disappears from the mesocosm and it becomes undetectable. However, we have hope that we can get enrichments of other Vampirococcus-like cells, and actually this has been the case by thanks to Ferial Buderka in the lab, who has enriched and is able to maintain in infected cultures new Vampirococcus-like organisms that appear to infect non-photosynthetic gamma proteobacteria. And they come from very different environment. In this case, it is from a freshwater shallow system in a natural park close to the lab where we work in France. And so it's not a saline or a highly sulfidic system. And we have been studying this type of system for many years, and we knew that CPR bacteria can be abundant in these systems. The 16S ribosomal RNA sequence of this new Vampirococcus-like organism is actually quite divergent from that of Vampirococcus lugosi, suggesting it might be even a new genus. But all in all, the fact that it also parasitizes gamma proteobacteria indicates some group level specificity and potentially coevolution, though it remains to be studied. So in summary, we have been able to describe a second example of extreme parasitism in CPR bacteria. Is it a general feature of CPR bacteria, or some of them can be simply mutualistic symbionts? It remains to be determined with more examples. There is a highly reduced genome, especially metabolism, but also many unknown genes that represent possibly new biological features that remain to be studied. And CPR are also very abundant in some environments, and they most likely play an important ecological role, and being equivalent to viruses and grazers in controlling the population demography and terminating blooms. So if you want to know more, you can go and read the article. And I want to finish by thanking once again the organizers of this NeLLi Symposium, David Moreira, who did most of the work the analysis, the phylogenetic analysis, but also other collaborators that work in this project, and of course the members of the lab for keeping a nice working environment. Thank you very much to you for your attention.
Thank you, Puri. Wonderful story. We have one question from Eugene Kunin. Eugene says, beautiful story. Have you detected any prophages? What do the CRISPR spacers target?
Yes, we don't really know. So they might target viruses, and actually in some CPR bacteria, viruses are known. But it might also be other parts of the host. We don't know. Possibly there is a bit of, but we have not detected any virus. If the viruses would be to describe, actually, they are unknown.
Okay, so I have a question too. So do you have the Lugosi in culture with the halochromatium? Are you able to maintain?
That's what I was saying. We kept it in mesocosm for a while, for a few days, but then it was impossible to maintain. It was a highly aggressive parasite, and in very few days, it had completely terminated the bloom, and it disappeared from mesocosm apparently. So it must be there somewhere. But we don't know which are the specific conditions under which it blooms. We are trying, but it's not necessarily very easy.
So have you tried it to prevent with a different food source, a different halochromatium species from a different place?
I think. So what we think is that there is a high specificity. So if there was another halochromatium species, it would be parasitized by another Vampirococcus. It would be very interesting to study virulence in these organisms. But unfortunately for that, you need either to have cultures or recurrent observations in the natural environment. And so that's what we are trying to do. But we are also trying with this new strain that appears perhaps less virulent. It is another Vampirococcus, and that we can keep in cultures by infecting new cultures with infected cultures, and it appears to at least be maintained for a while. So we have hope in this new strain.
Sure. Brian Hedlund just put a question in there. Great talk. Do you have any evidence of expression of the giant proteins? Do you think they're expressed as single proteins and potentially cleaved into smaller ones, smaller adhesins?
We would like to know, but we have no idea. No, we don't know because we cannot. We haven't any expression analysis. Yes. This is work actually from single cells and observations in the natural environment, or almost so.
Okay. Well, it looks like there's two more questions. One from Christian Rinke. Very interesting presentation based on the discovery that UGA encodes for glycine. Would you say that this stop codon recoding? And I'm not sure what the question is, although the UGA recoding for glycine was shown for SR1 and GNO2.
Yes. This is known in other CPR bacteria, and actually some people have proposed the idea that maybe it is a way to protect the cellular DNA from the foreign DNA that enters the Vampirococcus cytoplasm. Because the codon is altered, so the proteins, the genes coming from an external host would not be expressed correctly because they would be.
I just see that the Christian's half answer came after that, and it's basically about this evolutionary aspect. Dan Speth has another question. That the 1.3 megabase genome is relatively large for a CPR, and if there is any relationship between the genome size and the predatory lifestyle, as opposed to the more benign TM7.
Good question. I don't know. That would be interesting to look at. It's clear that there are many new genes and that we don't know what they are doing. And so they must be likely involved in this predatory lifestyle. Yes, we need more examples to correlate genome size with the lifestyle. And for the moment, there are not so many.
Actually, it's sort of on the similar aspect from the micrographs you presented. The size of the Lugosi is about 0.5 micron or potentially even larger. So it doesn't look like this organism fits the classical view that all these so-called CPRs are really, really small. This is, it's small, but it's not that small.
It is true, but also the host is bigger than the average bacteria. So yes, it's in proportion because these photosynthetic bacteria are rather large. Okay.
So if there's no more question, everybody's welcome to the breakout.
But thank you everyone for joining us. We hope to see you next week. And we'll see you all shortly at the breakout session.